Hello and welcome to our Lakeside chat. We are David Wofford and Katie Noonan, co-chairs of the Rotary Nature Center Friends. And we're so happy that you can join us tonight.
As we begin our program, it is important for us to acknowledge that we are on stolen land, that Lake Merritt is part of Ohlone territory. We hear now from Corinna Gould, spokesperson for the Confederated Villages of LaShawn and co-founder of Segura Te Land Trust.
Good afternoon, relatives. My name is Corinna Gould. I am the spokesperson for the Confederated Villages of LaShawn. We are here today at what most folks think of as Lake Merritt. We are in the territory of Huichin. Huichin is actually a territory that encompasses 6 Bay Area cities: Oakland, Berkeley, Alameda, Emeryville, Albany, and Piedmont. And this was a place of abundance. I'm so happy that people from all walks of life that now come into our territory can enjoy this beautiful place that my ancestors have enjoyed since the beginning of time. My relationship to the land that I have been born to, raising my children and grandchildren here, has been to tell the story, the truth of what happened on this land before other people came here. I'm hoping that as we begin to learn these lessons of fires in California, the pandemic that's happening, that human beings come back to living in reciprocity with the earth.
Thank you, Corinna, for a great framing of our program tonight. I want to welcome everyone again to join us for a show celebrating the plankton of Lake Merritt. As usual, what we're going to do is share a couple of our of really significant events for us this as an organization this month. So I want to share a couple of slides with you, and then we'll introduce Janai. In the trivia, we mentioned the City Nature Challenge, which was a big deal, and we organized people throughout the Lake Merritt area to come and search for and observe organisms, tiny ones like plankton and big ones like trees and mammals, fish, whatever, just to get a sense of what lives in the urban area that we inhabit. And it was a really fun day. Yes, so we turned out a number of the notable naturalists of our area. There's Janai with her microscope. We had Damon, Ty. We had Jim Irvin in our audience, I believe, today. Of course, me and David. We have a young woman from majoring in environmental studies at Cal Berkeley, and a young man is working on surveys of birds for the USGS. So they were wonderful to have on hand. We were able to bring in our volunteers from the Achieve group. These are high school students who have been with us all year, and they got busy making iNaturalist observations and looking in the camera. And as mentioned previously in the trivia, we turned up a fair number of species in Lake Merritt, including marine species that had disappeared right after the big fish kill in August of 2022. We have been doing surveys every few months of the same type as this to see what's alive, what's making it, and we were very pleased to see a full range of biodiversity of all types, and it was really a great event. We included some recreation for our young and some Seek stickers for those who participated. So it was a really wonderful day and representative of how Lake Merritt can bring people together and have fun and learn at the same time. That's what we're about tonight. I'm going to stop this share now, and thank you all for being here tonight. And I'm going to ask Janai. Are you ready, Janai? I'm going to introduce Janai in just a minute, but I'd just like to uh, go through just a couple of really simple housekeeping things that we may have already mentioned before in your experience with Lakeside Chats. But that the speaker is going to talk for a while, maybe half hour or so, and during that time, you're welcome to put your questions and comments and your knowledge into the chat. And Janai has said that it's fine if if a timely question comes up, just to interrupt her and let her answer it right on the spot. So do not feel shy about putting something into the chat. We're going to, at the close of Janai's formal presentation, we're going to open things up for questions and answers. And if you raise your hand and we'll go through the questions in the chat, you'll be able to unmute yourself and we'll be able to see you, and you can ask your question yourself in person. So that's how we are going to do that. Janai, are you going to be able to stay a little bit after 8? If we keep going?
I definitely can.
Okay, so we will be stopping the program just before 8 o'clock to give thank yous and credits. And then if there's more discussion, we're ready to go on a little bit after 8. So, so let me introduce Janai to you. Janai is an artist, a naturalist, and a enthusiast of the microscopic world. She is the president of the San Francisco Microscopical Society. And she just travels all over the place in pursuit of knowledge of things very, very tiny. Not only does she make beautiful photographs and drawings, but she knows a lot about these organisms and how they fit into the world and how they, contribute in their own way to our lives today. So let's see, mention a couple other things. Yes, she works as a interpretive naturalist with the National Marine Sanctuaries, and she works with any number of other, whenever she can, get out to spread the word and to share her enthusiasm for plankton and microscopic organisms and even little things that you can find. It's all surprising. So Janai, thank you so much for being with us tonight. And please introduce anything if I've left anything out. But it's such a pleasure to have you tonight.
Oh, thank you, Katie. No, you were very comprehensive. Wow. It's always a pleasure. I really enjoy spending time with the Rotary Nature Center friends and exploring Lake Merritt. Katie has opened my eyes to the plankton that's there, and it's been a really fun journey. So I'm going to share my presentation, which is actually really short because I want to get to the live stuff. So hopefully you can see my presentation now. Is it there? Somebody give me thumbs up. Yeah.
Oh, yeah. Oh, yeah.
All right. Thank you. So we're going to be talking about plankton, not just microscopic stuff, but plankton. Plankton's really my favorite segment of things that you can look at with a microscope. I'm going to fly through this a little bit. So please put questions in chat. And I think Janice or David are on duty to jump in and interrupt me. But it's possible I might get to one of the answers here for you. So when we're talking about plankton, one of the things that that I need to share with you is a little bit of vocabulary, because it's talking about things that aren't in everybody's daily life. I mean, they might be in mine, but not necessarily everyone else's, at least maybe that they're not aware of. So we're going to have some vocabulary tonight. We're going to learn a little bit about how you group plankton into different categories. Scientists love to do that. They love to sort things into different categories, sometimes based on how they live, sometimes based on what they eat. And then we're going to talk a little bit about why it's really hard sometimes to get a good identification of plankton, because a lot of times scientists will collect plankton and they'll look at something in it and you might want to know, you might see something. They're like, what is that? And oftentimes, to really drill down into the very fine detail of the species of that organism, it takes time and sometimes genetic testing. So we're gonna find a little bit about why. So here you can see I have plankton nets. These are some plankton nets that are setting out on a pier where I started collecting plankton on the edge of the bay near the Golden Gate Bridge. Here's one that's in the water. You drag it along in the water, kind of like a windsock to collect the plankton. It's kind of like a pasta strainer, except instead of pasta, you're collecting plankton. And depending on the kind of net you use, you can either get a golden brown, beautiful, rich phytoplankton, or you could get what looks clear but might actually be full of larger organisms that aren't photosynthesizers or don't have that same pigment. So to define what plankton is, right? Our first thing. So how would you explain what plankton is to somebody who may have never seen plankton before? I ask students this, and it's always interesting to hear what comes up, what their definitions are. So the definition, the functional definition that we're going to use is it needs to be alive, it needs to be in the water, and it needs to be a drifter. And by drifter, I mean not something that that can't swim at all, because a lot of the tiny things we'll look at can move, but they can't swim stronger than the current pulls them. So if the current's going from Oregon to Los Angeles, they can't decide Hawaii sounds good and take off that direction across the ocean and make it there. That's not in the books for plankton. They might accidentally get pulled on a current there if that happened. But mostly, if the current's going from the north down to the south, that's where they're traveling to. Let's see here. So kinds of plankton, I said phytoplankton, the golden brown plankton, they're beautiful. They photosynthesize, they make their food from the sun. And then there's the zooplankton, they're the animal plankton. They eat the phytoplankton. And there's all kinds of organisms that are in the zooplankton, the animal plankton. But these are some examples of those photosynthesizers, the ones that make their food from the sun, these tiny little nuggets inside this big giant cell are the part of the organism that makes the food from the sun. And this one it's less well resolved, but it still has that golden brown color. But then you can take the zooplankton and you can split them into groups. So there's some plankton, right? They're alive in the water and they're drifters, and they're going to be drifters their entire life. That's it. They're drifting now, they'll drift later. That's how they're going to stay in the water. Or they could be meroplankton. Over here, they're plankton only for part of their life cycle. Maybe they start out as tiny little drifters, and then later on they'll settle out, grow bigger, maybe live on the ocean floor, or they'll become really strong swimmers and they'll be able to take off and maneuver in a current and not be considered plankton anymore. And of those zooplankton, here are some of my favorites, and I've imaged these over the years. These are from the bay, so not necessarily from Lake Merritt, but they're all different types of zooplankton, and a lot of these are babies, and some of them are adults, but they start their life as little polyps stuck to something. Only this one here is plankton its whole life. This is the only hollow plankton on this page. So, but then how do we see them? So this is the sample I actually collected today at Lake Merritt. This was taken today at Lake Merritt. And this is my microscope. I'm gonna put a slide on the microscope and we're gonna have a look at what's fresh and zipping around. But without a microscope, you can't always see the plankton. Now, there are some plankton that are huge, like jellyfish, and other siphonophores and big things that drift around the ocean. Um, but what we're looking at today are the tiny ones. So in the definition of alive in the water in a drifter, I didn't say tiny, even though they can be, and there is a lot of the little ones. These here are all dinoflagellates, and these ones are organisms that we sometimes see at Lake Merritt, but some of them are more open ocean that end up being pulled into the bay. But there's a huge variety of shape just with this particular kind of plankton. Here's some more. I really, really love imaging them. These ones look like they have little armored plates on them. These ones here have, they have like a stinging cell, like a harpoon that they use to hunt other dinoflagellates. And we had these in Lake Merritt not that long ago. And one of the reasons why it's hard to identify them is these are all the same genus, but they're not the same species. So I would tell you these are Protoparadinium. It's a fun name. It looks like a pair of pants with a party hat on top to me. That's how I remember them. They're dinoflagellates, so they have a tail that's wrapped around a whip tail, like a flagella wrapped around that waistband-like structure, and one that comes out between the 2 points. But they're different species, even though they're all in the same genus. And it gets complicated. You have to figure out how the little plates align, how this little groove in the front aligns. So oftentimes you can get close, but not precise unless you do the work in the lab. And sometimes that's genetic work. So what is a bloom? Because there was a bloom at Lake Merritt. It was 2022, I think. I'm going to play that one more time. This was the Heterosigma akashiwo bloom that killed all the fish in Lake, not all the fish, but killed a lot of the fish in Lake Merritt. And this was a scoop of water. It was very, very heavy bloom. This is what they look like when they were all individually. I took a bunch of pictures, but they look like little cornflakes, little evil cornflakes. But one of the reasons why they're so hard to identify is not too long after you take them out of the lake, they start to die and they look like this, which is just kind of a clump. They start to disintegrate really rapidly. And preservatives that you would use to keep them also make them do this. So it can be a real mystery to be able to figure out what a plankton bloom is. So here, this is a little bloom. It's a little localized bloom that happened at Lake Merritt. Katie and I were out there and we're like, wow, the water is really kind of green and cloudy. And this is what they were. They were these little tiny organisms. They're like Euglena. They've got a little kind of red eye spot and a whip tail. But it could have easily been any one of these other organisms. So there was a mesodinium bloom, a little mesodinium bloom recently, where the water looked red along the shore, and that's this organism here. There's this one here that has that looks kind of like those evil cornflakes that did all the damage to the fish. But this is Prorocentrum, which is a different type of dinoflagellate. And I don't know if it produces toxins, and it was there one day and then dispersed. You can also have blooms of things like diatoms, these little chains here of these golden cells. And a bloom is a population explosion. So we've probably all heard about the super bloom of the flowers in California. Everybody's so excited and they like rush off to see all these fields of flowers and they love it. And then if you say there's a bloom in the water, everyone panics, which isn't fair because they're just as beautiful. It's just sometimes they can have a very negative impact instead of just looking pretty on a hillside. But sometimes they do have a positive impact. Sometimes they're food for other things and they need to bloom to a certain state in order to feed the other organisms in the food chain. So, and they bloom most of these little ones bloom by dividing in two. This is an organism called Noctiluca, and it's a dinoflagellate, and this is how they start to divide. They start to pinch off between the two cells, and then eventually there are two. And so the day before a bloom gets out of control, guess what? It's just a normal day because they can reproduce so quickly. They have a really short doubling time. So if you've ever played the game, I always get the numbers wrong, where it's, 2, 4, 8, 16, and it's that doubling game they used to have on phones. Anyway, once you start doubling very, very quickly, the numbers become very, very large. And that's how these little organisms do it. And another way they do it is they can ride out hard times. So dinoflagellates have these little stages where they're like seeds, but not seeds. They're like eggs, but not eggs. It's a cyst. It's a resting state. So they can hang out until conditions are just right. And when they are right, then they can start dividing. So there we go. But mysteries are real. Identifying plankton can be tricky. So what are we going to do next? We're going to look at some live plankton. And if you get an opportunity, look at what's in the water. I highly recommend it. Make observations if it's the color of the water, if it's fish you see eating something. I think it's all really important. And then share that with someone else. Sharing what we learn, I think, is very important. So I'm stopping my share. And now, if all goes well, I am going to put live plankton. I'm going to shrink myself up into the corner here. If you could pin me for folks, I will make a slide and put a fresh sample. And if anyone has any questions, now's a really great time. Any questions, Jana? No? All right. Well, we'll see what we've got here. I'm going to move this slide here out of the way.
The whole hanging out until conditions are right for splitting and reproduction,
Yeah.
multiplying. Is that
Yep.
what you consider that to be fairly common in nature, or hit and miss?
Well, we can't do it. Mammals can't do it. Birds can't do it. Most of the larger organisms can't do it. It's like a cross between having a seed and an egg. And it's different from an egg because you don't have to, like when eggs have a certain incubation period, you can't like hang out in the egg state for a really long time. Typically, if you're a bird or a reptile, right? There's an incubation period, a period of time where you can exist as an egg and then you have to hatch, right? Like time's up. But dinoflagellate cysts, diatom cysts or resting cysts can exist in a body of water for a very long time. And I don't actually know if anyone's done experiments to find out what that time period is, how long they can persist and still be viable. Like how long does it take for them to not be able to kind of pop out when conditions are right? Because a lot of times we won't see those organisms. They won't be in the water. I mean, or if they are, they're very rare. Here's a little diatom. I'm going to just see what else we can find. And so
Let me know when it's a good time for another question. We've had one pop up a bit.
Oops, let's see. I'll try and get that one in focus. So they can move pretty tracking them down isn't always, there we go. So that's a Nauplius, that's a baby copepod. It looks like, or a baby barnacle. We'll see if it turns sideways and we can find out. What was the other question?
Well, it has to do with viruses.
Oh, go ahead.
And even though most scientists don't consider them to be alive, some do.
Mm-hmm.
And let's see, they were listed in our chat slide
Yeah,
tonight. So what I guess what's your piece the different types and how that plays out?
yeah. So that's interesting because so one of the things, it's always a good question,
I
right?
want
I
to say
love,
that question is from Ingrid, by the way, but go ahead. Yeah.
yeah, thanks, Ingrid. Um, so the idea that of if you agree that viruses are alive, then viruses can be plankton because they exist in the water column and they impact organisms. There are viruses that attack prey on. I don't know. It depends on how you categorize them. And I don't study viruses, but it's always a great question because it's about being alive, right? So if you consider them alive, then you will also need to theoretically consider them plankton. There's bacteria that's plankton. There are fungi that are plankton. Diatoms and dinoflagellates. This is a beautiful little worm that keeps going in and out of focus because it's swimming up and down. And my microscope has a very narrow field of focus. Let's see, there we go. Oh,
yeah.
sometimes they slow down. Sometimes they don't.
Oh, yeah.
Anthony wanted to know the question, is photosynthesis the process that causes the bloom,
So
where they multiply many as
yeah.
a result of warmer temperature and increased sunlight?
So photosynthesis is how they eat. They eat, they're sunlight eaters. They feast on the light from the sun. And so in order to, let's see, let me just find something here I can focus in on for us. That one diatom that's sitting really still, we're going to zoom in.
Oh, yeah. Oh, yeah.
And so when they photosynthesize, that's how they eat. They use the sunlight, the energy from the sun, and then they take up the carbon dioxide and the carbon from the environment and other nutrients, and they build more of themselves. So if there wasn't sunlight, those particular organisms that use it to make themselves, to build themselves, they wouldn't be able to bloom because there wouldn't be enough sunlight, which is why we often don't have blooms. We don't have this like either flower blooms or plankton blooms in the winter, at least in the northern hemisphere, because there's less sunlight, less hours, less days of sunlight, or clear sky with no cloud cover. So it is one of the things that enables them to do that, but just because it's their energy source or their food source.
Oh, wow. Jim has identified the worm larva as a spionid worm larva.
Yeah.
Thanks, Jim.
Those polychaetes are beautiful.
They are. So I wanted to go back to the virus thing because
Yes.
it's like a good question, and I'm going to put this in the chat letter when we put it out. But apparently, viruses do infect and prey on some of the phytoplankton
Yep.
and the dinoflagellates, and some people are even looking at that as a possible biological control approach, or just even to understand what they're what a role is, because we don't, we even under the microscope, we do not see them. So
No, no.
it's a really interesting question is where they fit in to the ecology.
Yeah, so they already fit in. I'm
Mhm.
going to switch slides here. They already fit in. They're already present in the environment, and they have very specialized relationships with organisms that have been around, the relationships have been around for a long time. How we are learning to understand them is a whole different thing. There's some more of those spionid worms for Gem. There's some really lovely worms. See if I can. So this is dark field that we're using right now, and trying to keep everyone in focus. I am going to switch to a different mode of
Here goes
viewing.
an article.
Yeah, they look like little little shooting stars in the dark field. But I'd like to switch to a different mode of viewing. We're going to zoom in. And when I do that, give me a moment. It's going to go bright, sorry. And then we're going to go here. So hopefully we'll get one that slows down just a little bit, and we'll be able to see a little bit more of the details on it.
So Beth wanted to know how can you tell if it's a phytoplankton or a zooplankton? Is there yeah.
So size is part of it. And then there's an entire group of organisms where it's a little sketchy, where the traditional definition, right, that we like to group things in categories as humans, we sort things. However, the things that photosynthesize, that make their food from sunlight, are considered phytoplankton. And then things that eat other organisms like us, whether we eat plants or we eat animals, we're eating something else. We don't make our food by standing out and holding our leaves or hands out. Using the sunlight, we don't get that energy. We have to get our energy from what we eat. So the zooplankton do the same. The zooplankton have to get their energy from other sources. Oh, see, there we go. Oh, and it goes. This is what happens when you're looking at live plankton. They sometimes they cooperate, sometimes they don't. I spend a lot of hours looking at them. So the thing is, there's this interesting group of organisms that are in between, where they photosynthesize and that one's spinning really quickly. They photosynthesize and they eat other things, like the dinoflagellate. It's going to slow down in just a moment, I hope. It looks like it's stuck on the oh, it's not. It's just spinning. Sometimes they get stuck on the cover slip with the tension of the water, but it's.
Janai, I have to say, you're disturbing my equilibrium at this point.
Oh no.
For viruses, because I've always, in my head, they need a host, and they're not alive because there's some systems that they just don't do. They don't create their own energy, and they those are the things that are in my head. I need to think a different way, I guess.
Not necessarily. Like so, the definitions of viruses and prions and all of these other things is evolving, and I don't study them. So the best I could say is read and find out, seek out people who know more about it. But the thing is, if you do consider them alive, like a cell parasite, like depending on how you define them, there's potentially a lot out there. But that's just one piece, right? I mean, I don't see, you can't see viruses.
Mhm.
And bacteria are notoriously difficult to see.
Oh, yeah.
But these organisms are much bigger. So they are, these are zooplankton for certain. And in fact, they're in that fun category of meroplankton because they don't stay in the plankton their whole life. They're only plankton for part of their life. So they're here because in the plankton, because that's where the food is. They're swimming in a food soup. And the food soup is made up of things like these little chains of diatoms here, these little golden nuggets that have cell walls of opal. It's a biogenic silica and it's amazing. I'm going to try and grab another slide. I'm going to zoom out a little so we can maybe, oops, sorry, bright light. My camera has a little bit of a lag when it comes to adjusting, so I'm sorry. Let's see. And we can get them.
Janai, Anthony is asking about where an algae bloom like the red tide, I think probably referring to the Heterosigma aquachuo bloom in 2022. Where does that fall into the scheme of things? The idea of a red tide.
So red tide, when you say a red tide, you're basically using old terminology because people used to see color changes in the water, and then they would note that things happened after the color changes in the water. Maybe animals got sick or fish died. And so they would call them red tides. We have learned now that it doesn't have to change the color of the water to have a negative impact on people, animals, and the environment. They can cause harm without having that visible color change. And certain organisms do it. In fact, there's a diatom that does it. It's called Pseudonychia. And Pseudonychia is responsible for something that's called domoic acid. And that causes amnesiac shellfish poisoning. So sometimes they close the shellfish season. Here we go. We're gonna zoom in on this because these are actually quite beautiful. There's a lot of the phytoplankton in this sample. Oh, and there's a rotoplasm.
Did that last reference mean to say that the shellfish loses its memory?
No, but if you eat contaminated shellfish that have been feeding on the organism that creates the domoic acid, then you could get sick and have memory or neurologic issues. There are sea lions that'll get sick from eating things during a domoic acid event, and they'll have these like repetitive behaviors where they'll like go in circles, and there are other problems. The Marine Mammal Center tries to rehabilitate them, and sometimes they can, and sometimes they can't. And it doesn't change the color of the water, even though they're present in the water. So red tide it's just an older term because it was something that you could see. It was a person scale change that allowed people to notice when something changed in the water. But let's see here. I wanted to show, I've got my arrow here. These here are diatoms, and there are a lot of diatoms in this sample. That is a dinoflagellate. See it swimming around? Hold on. There we go. If I can get it, it's swimming up and down around these diatoms, so it's not holding still. I'm trying to, oh, there it goes. I can almost get it into focus. But all of these diatoms, they're drifters. And they drift in the water, but they can also, they've been doing a lot of genetic work and they've been studying them, and they find that the diatoms can also affect where they are in the water column. So even though they're drifting, if you think about how someone with a hot air balloon navigates, where they can move up or down based on the current of air to help them get where they want to be. Here we go. See if I can get this a little bit closer. So these diatoms they're a catastrophe. They're a really interesting diatom. And they've got these funny little spines on them. And the diatom uses that to help it stay in the water column instead of sinking down too far where the sun can't illuminate it. Let's see if I have to kind of rotate through some of the settings here. Here you can almost make out little shadows of those structures that help, kind of like a drag line, but they're soft. Go ahead.
I would imagine that those diatoms are made up of a bunch of individuals strung together.
They are. Each little nugget, there's something nearby that's causing them to do that. Each little nugget along this chain is an individual cell. And this chain here is a chain of clones. They're all genetically identical.
Well, I could see how once you became a part of a structure like that, you could manipulate yourself through, bend and sort of respond to the current in order to change your direction, up or down.
You can.
But can you do that as much as an individual?
Well, it's a strategy. There are different diatoms, and they evolve different ways, different strategies to stay in the right spot. So like here's a straight chain. These are called skeletonema. And they hold together with these beautiful tiny little threads of hydrated silica or opal. And they can stay adrift. They're very, very tiny. And they don't have those extensions, but they have the extensions between the cells. And then over here you have these catastrophes. And if I get the focus just right, you can see these long hair-like strands that kind of come off them. And that photobombing us here is a rotifer. Oh, no, there it goes. Here's a dinoflagellate. There's another little organism drifting by. Here's another catastrophe. You can kind of see it a little bit better, all those little extensions. So that's one strategy. Let's see if I can. This one here has a very large cell size. It's kind of covering itself in junk at the moment. But one of the other strategies for the larger cells is when they photosynthesize, they can make like fats or lipids, right? And that can help them because they can change their density or how they float in the water. So there's a lot of different strategies they've evolved over time to be able to stay where the light shines, because where the light shines is where they can make their food from the sunlight. Let's see. I really love the phytoplankton. There were some triangle ones earlier tonight. I'm going to move the arrow over. Any other questions?
I should be looking in the chat.
Ooh, there's the triangle one I was looking for. Here's another.
Yeah.
Oh, wow.
So this one here, it's being moved by another organism there. That's either a baby barnacle or a copepod. Let's see if it'll hold still for us. No. Oh, this is a tintinid. Sorry, it's a little bit like whack-a-mole. When I'm looking at the plankton, I'll see something and then it'll swim away and then I'll find something else and I'll focus on it. So this here is a ciliate. It has a hard lorica, which is like an outer shell. Ooh. And give them a moment to settle down. And the tintinid hoovers up little tiny things. It eats really small organisms. It's also a single cell. So the things that it eats has to fit inside its mouth. Things like bacteria and little bits of broken down diatom or dinoflagellate. Who's that? There was another. There. Oh, look at that.
Oh,
Oh,
beautiful.
it's a dinoflagellate just swam by. Did you see? I wish I had a rewind, but there's no rewind on the microscope. There it is.
Okay, I'm going to say this because I'm looking at the chat and I'm missing all the pictures. It says, can you please tell us something for size reference? This is from Joy, such as the width of a hair, whatever, for these planktons we are looking at. Thanks.
Ah, wonderful. They're dust sized. So like the size of the dust in your house is the size of most of these. The very tiniest ones would be the tiniest specks of dust that you only see if there's a lot of them built up on something. The largest ones are the bigger specks. Sometimes I liken it to when, if you've ever been sitting someplace really still and there's some dust in the air and you see a ray of sunlight coming through, and all of a sudden the dust that you don't normally see look like these little bright sparkly specks in that ray of light. That's the size of the majority of these organisms. Hair is a hard one because it really varies in thickness and coarseness and personal experience. Sometimes, maybe at the end when we're wrapping up, ask me again, and I can stick one of my hairs on the microscope, and you can see what a particular hair thickness is. But so rotifer, this one that kind of looks like the flying ice cream. Let's see. I'm gonna zoom out of. It keeps turning. They're lovely little organisms. I'm going to zoom out one. It's going to get a little bright again. I'll have to adjust. But here we go. We can maybe get that rotifer in focus a little better. There. Oh. So the rotifer, this one that I'm trying to focus on here. Let's see if I can get it. It's just moving too much. They're amazing little organisms. They are they're a great group. They're plankton their whole life. So they stay plankton their entire life. Oh, and there's a dinoflagellate swimming by. Sorry. It's a really beautiful sample. So this is for me, from what I'm seeing, it's a healthy, diverse sample at Lake Merritt. There's a lot of different sized organisms. There's a lot of different kinds of organisms. There's plenty of the phytoplankton, the zooplankton are zipping around and they're eating. And it's really lovely to see. Sometimes I've looked at Lake Merritt and it's just like when there's a bloom and it's just one thing. So it's really nice to see the variety. There's another, that triangle one is called Lichmafora. So it's a beautiful phytoplankton sample.
So Janai, we have a nice question here from Anthony.
Yeah.
Besides predators of plankton, like other zooplankton, fishes, and even whales, what other environmental factors can have a negative effect on plankton?
Oh.
That's a big question. Are there migration changes? What about climate change?
Oh my goodness.
Yeah, that's a good question.
It's an interesting question, and there's so much to it. Um, so there's a lot that we don't know. I'm going to just start by saying that. There's a lot that I wonder about. There's a lot of research that started asking that's been asking questions, but there are so many more questions that the research brings up. I had just listened to a lecture the other day that was about not viruses, sorry, fungi that attack particular diatoms and have a very significant impact. Like I can't remember exactly what the number is, but like it was a catastrophic effect on this one particular diatom in this one particular lake. And but they haven't studied fungal diatom relationships in a lot of different systems or with a lot of different fungi and diatom pairs. It appears that this particular fungus, this chytrid fungus, was only targeting this one diatom, and that was the relationship they were exploring. And as far as I could understand it, it was fascinating. So that's just one thing that could totally affect it. And they found that it was attacking the diatom during one phase of the chytrid's life cycle and only during light hours. So it had something to do with the photosynthesis process. And there were so many more questions they had, but it's not easy to find funding to research something. It's like, so who cares about Asterianella in a tiny little lake somewhere? It's not it's not it's not a financially viable crop. Nobody eats Asterianella. It doesn't necessarily affect like the commercial fishing industry in that area. So we tend to do research on things, not always, but we do tend to research things that impact us on our scale. Science for science sake is often more difficult to find for them to find funding. But there's a lot of questions. The virus interactions with diatoms, if it interrupts the diatom's dividing life cycle and allows the silica to stay suspended, how does that affect other types of diatoms? It's a huge question. Sorry, I'm like rambling now. This is an egg. I just thought I'd share this beautiful egg. We're going to zoom in on it. One of the other things that can affect populations of plankton, pollution can either encourage them to multiply, or the pollution in the water can keep them from being healthy and dividing and doing what they need to do. The change in temperature of the surface water can affect when things bloom, and that those things that bloom can be food that's important for other organisms. So I don't know if everybody here does, but people like to eat food from the water. We like to eat fish and crab and clams and oysters and mussels, lobsters and shrimp, and all of these things either start their life as plankton or eat plankton almost exclusively during one part of their life cycle.
Oh.
So if you think about all the changes that we're making to the waters, it has an impact on things that that we do interact with on a fairly regular basis, as in food. Sorry.
We have one question in
Go
the
ahead.
chat that asks what the impact of acidification in the ocean could be on plankton, but perhaps we should do our close and answer that afterwards.
Oh,
Okay.
oh my gosh, I didn't realize we were there already. thank you, everyone. We're going to take just a few minutes to show our closing slides, and then we can continue the conversation here. Thank you, everybody, for coming and enjoying this magical view of the plankton in Lake Merritt. Special thanks to Janai Southworth, also to iNaturalist, the Lake Merritt Boating Center, the Lake Merritt Institute, Adopt a Spot program, our Achieve volunteers, Major DD, and the Climate Polar Action Fellows, and all of Oakland. We're so happy you could join us, and we'd like to note that coming up, we are going to have a chat on June 7th with Eli Kirsch of the Lake Tech Corporation, talking about putting oxygen in Lake Merritt. Eli is the president of the Lake Tech Corporation that is doing some monitoring, giving an incredible amount of 24/7 coverage to water quality conditions in Lake Merritt, and also piloting some infrastructure to possibly change and modify the amount of dissolved oxygen available in Lake Merritt. So it'll be a really interesting discussion. Coming up in June, right down on the shores of Lake Merritt, we'll be participating in another California Academy of Sciences event called Snapshot Cal Coast, because of course Lake Merritt is really just a section of the coast of California, a little pocket of the ocean coming in off of the bay. So we'll have another table, another great place to be able to come out and see the plankton in real time. We'd like to thank the KTOP Channel 10 TV of Oakland for rebroadcasting our programs. So our programs are broadcast every Sunday night at 6 p.m. to 7 on KTOP. The first 2 Sundays of the month show a collection of older programs, and then the last 2 will show the current program. So it's a great way to check in on the program on TV. And then we'd like to thank everybody for being here, and like to mention that our mission is that to advocate for the Rotary Nature Center in Lakeside Park as an interpretive education and science center for all the people of Oakland, and as steward of the Lake Merritt Wildlife Refuge. If you enjoy our programs, please check into our website. We do post the next Lakeside chat links and also other links. We want to thank Rob Lamone, our producer, and we would like to thank thank you to all of our volunteers and our participants in our programs. We would like to give special thanks to the Frederick E. Hart Foundation for Educational Opportunity that has helped us to keep the Lakeside chats going every month. So we're really really thank these people for contributing. We thank you for coming, and if you do like what we do and would like to make a donation, you can do so online through our website. This brings our program tonight to a close, and we want to thank you for being here and encourage you to check in again next month on June 7th, when we're going to have another really fascinating talk about Lake Merritt. We can continue our conversation. Thank you so much. Oh, wow, look at that.
Yeah, if you wanted to pin me.
That I could do. Okay, just one moment, please.
Somebody asked about ocean acidification and what does that have to do with the plankton, and I found this critter here. So this is a baby clamor mussel. It's very, very young. And one of the things that you might notice is it's so small, its shell is basically transparent. You can see through its shell to its internal organs. That's not stuff on the surface that is making a pattern in there. That's actually, oops, going to start spinning around. They're called veligers when they're tiny like this. This is plankton. That's plankton when it's little. It's marrow plankton, and then when it grows up, it'll settle down. But their shell is so fine and so delicate at this point in their life cycle. A small change in the pH, making the environment just a little bit more hostile, will mean it has to use more energy to build its shell, and it can either keep it from growing at all, so that it won't be able to get big enough in the right season to be able to settle if the water conditions aren't right. It could impact it where it might become easier to eat, so other organisms would who might not have eaten it might be able to eat it. Can make them vulnerable to illness, disease, bacteria, if they aren't in an environment that's healthy for them. And for it to be healthy for them, it needs to be not acidic. And I don't mean like not lemon juice, just a tiny shift in the pH can make a huge difference when you're this tiny and you depend on the environment for basically your life support. They're really quite beautiful. This one's not holding still though.
Still pretty beautiful.
They are in their own way. But it's spinning right around. So that one, that diatom that it's spinning around, that chain of clones, that's called skeletonema. And there's a lot of it in Lake Merritt, and it's one of my favorites. They're very tiny. Ooh, there was something else that just swam by. But if you ever get a chance to look at them under a scanning electron microscope, I can try and see if I can find an image of one. They look like something that was sculpted by elves. They're very architectural. They look like I don't know what the word is, like a gazebo kind of. The structure between the cells has that, like a very ornate gazebo. And that one's just spinning in circles. All right, let's see. I'm going to zoom out and see who else we can find here. Maybe I'll just keep it there. Were there any other questions? Because I can ramble on a bit, and you can unmute yourself and ask at this point. I don't think are we recording still?
Oh, yes.
That's okay though. It's okay to record the discussion. That's kind of interesting.
Okay.
So, yeah.
So either unmute or you can continue putting them in chat if you're not feeling.
Yeah. If you want to ask a question, raise your hand and then you can unmute yourself now.
Nice. Thank you.
In the meantime here,
Yeah.
I was wondering, I would have thought that I would see in these displays more microorganisms that are not plankton, who are in the water and can move on their own accord and swim about and probably prey on plankton. Is there a reason for that, or am I mistaken?
So most of the stuff that I have here is so tiny, and I collected it with a net. So because I collected it with a net from the edge of the lake, the majority of them are going to be plankton. And the things that would eat this are bigger. And I actually had a baby fish in my net, in my sample jar, and I put it back in the lake. I didn't take it with me because it was almost a centimeter long. And I try and not keep anything large, that even if it gets pulled by the net. So So, I mean, there's detritus, right? There's clumps of stuff. I say detritus, it's the fancy word for junk or dirt. But let's see. There's a lot of it. In the ocean, it's important. Go ahead.
Ingrid wanted to ask, her mic's not working, but she wanted to ask why and how that organism was able to spin around the other stringy organism. And could you rename the two organisms?
Certainly. So the one that was spinning in circles around, that was moving really quickly, it's either a baby clam or a baby mussel. It's a baby bivalve, right? It has the two shells like a clam or a mussel does. I don't know which kind. They're hard to identify when they're tiny. You have to measure them and compare the outline of the shape, and but they have an ability to swim. They use a structure called a velum, which is like a membrane that they have a bunch of little, let me see if I, there we go, a bunch of little cilia on, like wiggling hairs. And so they take that structure and they can poke it out between the two shells, and they can swim with it, or they can pull food into their body with those little wiggling hairs. So they're veligers when they're tiny like that. And it was spinning around a diatom. It's a chain diatom, so it's in this long chain. And it was a whole bunch of cells. And I think that particular diatom, not this one, but the one before, was called skeletonema. And I'm going to try and I want to find let's see if I'll find one. Oh, look at that. Sorry. There's bacteria that's in here. It's been sitting. Let me get my arrow. Let's see. See the little tiny movement right along here. So this is an empty exoskeleton of, I don't know, probably a copepod. And you can see any little bit of its body that was left behind, any material that was left behind when it molted and grew bigger, is being consumed by these bacteria. And then things like rotifers will eat the bacteria. It's probably actually one of the more interesting bacteria images I've seen. For me, I'm not a big bacteria person. I mean, they're interesting, but like compared to little worms and diatoms, not so much. There, this is part of that same exoskeleton. It looks like you can see the very fine feathery structures. Let's see if we can. Oh, there's the skeletonema. Here, let's find a better one. Any other questions while I'm looking for the skeletonema? I'm easily distracted, so.
I thought, Wendy, you look like you want to unmute yourself and ask a question. I don't think she can unmute herself.
Oh. Let's see if I might be able to zoom in on this enough so you can see what I was talking about. I'm sorry if you can't unmute yourself. It's possible you can, you could type it in if you can't unmute. I'm going to try and zoom in on this. So the skeletonema has these fine little threads of opal, this iogenic silica. It's like opal between. So this is one cell, and this is another cell, and these are those tiny little threads in between. If I focus just right, you can see how they almost look like it's a little bit brighter here. That's where they're holding on to each other, where they like are holding hands almost like little knuckles. And if you could see it, if we could go closer, which we can't on my microscope right now, it would look like little skeleton joints. in between here. And there's called skeletonema. And that's the type of diatom that it was spinning in circles around.
Janai, we have two size-related questions here.
Yeah.
So Ingrid was noting that the movement of these little chains and the plankton seems kind of dramatic under the microscope, but in reality, they're just moving in a drop of water, and in like on a larger body of water, they probably would not be moving that much. Is that more or less correct?
So they would be moving the same amount, whether they're in the drop of water or in
Yeah,
a.
that's, I mean, I didn't put that right. Yeah.
Say,
But they would not, yeah.
But so the field of view is what you're talking about, like how, like the way they're moving here. So I'm going to zoom out to give you an idea. Let's see. This, let me see, go one more. Here we go. Sorry, when I switch between modes, that skeletonema gets really, really tiny all of a sudden. So this is about 2 millimeters across, 3 millimeters across, I think. Let's see. I was just going to try and put a hair on a slide, and I lost it. So at my lowest power magnification, the entire width of my screen is about 4 millimeters across. So this one here is close to like 2.5.
How many millimeters does your string of hair average?
It's less than a millimeter. That's why the thing is, right? Like average, I'm really, so math. See, I'm doing visual stuff because math is not my forte. But I can take a hair here and twist it up and put it on a slide with some seawater. And then we can all look at it together and you can tell me what you think. One thing you can do is if you have a calibrated camera, it'll measure for you. However, I've been using this camera back and forth between different microscopes, so it's not currently calibrated. Otherwise, I could just have it tell me, which is what I would normally do if I were, let's see, there we go. Cover slip, hair, and we're going to see the hair experiment. Here we go. All right. So let's get the hair in the field of focus. There's the hair. You can kind of see the scales on the hair. So that's how big a hair is, or 2 strands twisted together. And that's one of those little worms. Oh, right. Just to give you an idea of, I have a polarizer on, so the hair's looking particularly rainbowy tonight. Right, I'm going to zoom in. Right, so this is we're looking at this magnified approximately 100 times if you were looking with your eyes through the oculars of the microscope. And then this is approximately 200 times. There's the little worm compared to the 2 strands of hair. It'll swim out in just a second and then we'll poof. That's actually kind of cute. And then where we were when we were looking at the really zoomed in, we were here. You can actually see the scales on the hair. Let's see. I can try and adjust this a little bit so less rainbowy, maybe. Sorry for the psychedelic adventure. I'm playing with, there's a crystal that allows me to see features in the microscope that you would normally have to stain things to see in my microscope. So I can look at live organisms and see certain details. So you can kind of see the scales on my hair. I don't want to look at the ends. Don't look at the ends. There we go. There's that worm. So this is approximately 400 times magnified, and that's usually as close as I go when I'm doing this, when I'm sharing. We could go closer if we were just looking at things that hold still. But
Well, that worm looks like a born show-off.
yeah, it was showing off. And there's that skeletonema, right? That tiny little strand of diatoms compared to the hair. There we go. Does that help for scale?
Yes.
Wonderful. Actually, the hair's kind of cool. All right. And it's hard because it's not human scaled, right? Like we're so used to things on our size scale. Once we start looking at things um, that are on a different scale, different size, whether it's things that are huge and far away, like galaxies, or oh, that was a copepod. Come back. See, now I'm so zoomed in. There it is. A lot of looking at plankton is just patience and exploration because they don't show up on cue. There are things we can do to help find them with light and with the sample, but yeah, they don't. They often don't show up on cue. Let's see. Any other
Ingrid,
questions?
Ingrid wants to know, do you take photos of the plankton?
I do. Kind of obsessively. You know how people were into that whole Pokemon thing? I don't know if they still are, the like where you go out for a walk and you like catch the Pokemon in different places. I'm like that with plankton. I collect plankton and I look at it because I never know what I'm gonna see in the plankton. I'm gonna zoom out so we can get um, let's see. So now we've got some color here. All right. Where'd that hair go? Go this way. There it is. So I'm using the wrong objective for this style of so there's different types of when you have a microscope. Actually, I bet I could turn on my other camera now that we're in the after party part of the talk.
Yeah.
Here, let's see if this works. There's my microscope.
Very nice.
And so I had a picture of it in my talk so that people could see. But the different objectives, which are the part that does the majority of the magnifying that are close to the slide, they're designed for different ways of shaping the light so that you can see the light, you can see what you're looking at. It changes the way the light moves. And so some of these objectives are designed to have a very different, let's see if I can switch it out and I'll show you. This is phase. It has like little halos kind of around what you're looking at. It's a different way of viewing. But oh, I don't remember where the slide went that had the bacteria on it. But if there's a lot of bacteria, the bacteria stand out because there's little halos around them. But it's not as good for seeing other types of detail. So some of my lenses on my microscope are good for this, but then I have another one, and if I switch to it, this one here is called DIC, and it's very good at seeing structures that you would normally have to stain something, like make a special preparation to be able to see. The problem is when I switch back and forth between the different methods, there's a little bit of tweaking and fiddling that needs to happen. So that's enough of that. But you never know what you can find in the plankton. Any other questions while I'm scanning? I'll switch to a different slide. No? Okay. That looks really beautiful with DIC. There's things, there are features we can see in this organism using this type of illumination and magnification that you wouldn't be able to see with the other styles. Copepods are plankton their whole life. They never grow out of it. But these worms will. Let's see.
They're plankton their whole life. They never go out of it.
Yep.
Meaning that they never stop being a drifter.
Correct. So even though they can swim around up and down, I'm zooming out so I could change the slide.
Yes, I understand now.
There we go. All right. Let's see if we can switch up to another slide. Should we try one more slide, or is everybody planktoned out?
I think one more slide.
I'd like to see another slide. So Janai, what you're talking about, these are meroplankton. Where do we see them when they're grown up at Lake Merritt, some of these, or do we?
Oh, so on your settling plates.
So they settle down on hard structures, and I think some of them like build mud tubes around themselves, and others, I think some of them can probably burrow. There's some kinds of worms that burrow.
I mean, there are as many. So there's clams and mussels and barnacles. Mm-hmm. There's crab, right? There's all those different ways of being. There are shrimp, right? Arthropods, like beach hoppers. There's so many things that can start one way and then end up living in the lake in a different way when they're adults. All right, I'm going to try and make a mixed slide here. So we'll have some phytoplankton and some zooplankton. Let's see if I can get some from right there. Hopefully it'll be a nice mix for our last slide.
Yes, I'm sure.
Yeah, you never know. I mean, it's kind of like I was saying, like with Pokemon, it seems silly, but once you start looking and realizing how much is there and how many different kinds, right. Let's see here. This is bright field, and I don't have the white balance set. Let's go. So this was what I was talking about with the one of the modes of seeing. So this is called phase, and or phase contrast, and there's like a lot of little halos, and it works really well for some things, but not for others. And I don't have the patience to adjust it right now, but I really want to see. There we go. I like this particular mode. And we're going to see what we have. So we have a lot of those little worms again. Little polychaete worms. And the tintinids. Here, we're going to zoom in right there.
Wow.
I know, it's a lovely sample. Things are looking really good. So here, where's my little arrow? What I'm trying to see right now, this organism right here is called a tintinid, and it's a single cell. It has a hard part that it builds out of things around it called alorica, and it's getting buffeted too much to really get a good look at. So we'll see what else we can find. Oh, there's an egg. You can see movement inside the egg. Let's see, we can go in closer. Bring it back to the middle. There we go. So if you look carefully, so a lot of these tiny little organisms, when they're young, it's getting buffeted. But we got a little bit of a look. You could see that's actually the egg of a rotifer. You could kind of see the cilia inside of it. Hold on, where'd it go? It just got bumped. Well, it'll turn up again. Any other questions while I'm poking around here? There's so much to say about plankton, it's hard to stop it.
I had a thought I wondered earlier when you were talking about the creature that photosynthesizes and seeks out prey for nourishment,
Yeah.
whether or not any larger creatures have been known to do that to some extent.
Huh. So there are organisms that I think that they like there's like the Venus flytrap, right? So carnivorous plants do it, kind of. Don't know if it's really called hunting, but
There are a lot of sea creatures that actually capture the chloroplasts of some of their prey organisms and wear them, and then get the carbohydrates that are produced by those chloroplasts and use them for themselves.
kleptoplast.
Yeah,
Chloroplast.
yeah. No. They take the chloroplasts and they are the kleptos. Yeah.
Yes. And they're the chloroplasts that they're using aren't their own. Like
right. Yeah.
Here.
Pretty amazing. I think there's some bivalves can do that.
Katie, ecdysis, right there in front of us.
Oh no.
It just escaped from its.
Oh.
There it is. So it wasn't here, and now it's here.
Oh yes. It came out of its capsule, right?
It totally did.
Oh wow. Yes.
Sorry, we've been talking about this because we often find these empty theca, these empty hard parts, and then this dinoflagellate is slowly taking shape, right, of its new theca as it swims away. But it's hard to identify them sometimes because they'll do this, right?
Yeah.
Right? Here's the hard part from this one, right? Where it's got these little armored bits. And here it is now. It hasn't yet hardened its armored bits. And then what's going on here? I don't know. I mean, it looks like there's two dinoflagellates inside of some type of capsule. I don't know. I love that I don't know. I think it's so cool. I'm going to have to look that up now. Let me see if I can take a, let's see. Let's see if it works.
Janai, in this process, if we could draw to a close.
Yeah.
Yes.
You want to draw to a close? thank you for hanging out in the plankton. There's so much to learn. I'm actually trying to record this right at this moment because I've not seen this before. This here, I just think is
Can
really,
can I read a poem that
please,
Anthony just posted?
please.
The ocean is no place for a plankton. Its insignificance is immense. For in its end of days, 2 million more there shall be, and its demise will not halt the routine of the sea. Even it will not falter when its very shadow is banished from the earth. Yet the plankton considers the universe, the space between the worlds, is vast and empty. The stars that encapsulate them are but a dot on a celestial canvas. The journey of the shooting star is futile, yet it briefly paints the sky in a hue most beautiful. And knowing this, the plankton allows the current of the sea, the stars, and the spirits to carry him on his journey far and wide. The ocean is no place for a plankton, yet without him, an empty place it would be.
Wow.
Thank you, Anthony.
Thank you. Thank you, everyone, for hanging out in the plankton. And oh, there's another one. I have so much more to see here. It's always such a pleasure.
Janai, are you still doing the
Just like a plankton.
microscopes and chill at all? That people
Yeah,
they might
yeah.
want to know where if you enjoy doing this. That Janai leads a whole. Well, tell them about it, Janai. About
Tell us
playing.
about it.
Yeah.
So, I, in my wearing my San Francisco Microscopical Society hat, we host events and volunteer places on a fairly regular basis, and we have very informal gatherings that are kind of like hangouts. We do one in Berkeley at Triple Rock Brewery, and we do one in San Francisco at the Valley Tavern. So there's something happening kind of every week. You don't have to sign up. There's no lecture, there's no talk. You kind of wander in, and people are usually there sometime between 5 and 8 p.m. is the time frame. But it's meant to be a time and place for people to nerd out about microscopes and microscopy and support each other in their habit and enjoyment and their hobby. Yeah. So we do that.
That's so
The
cool.
next one is Wednesday. This coming Wednesday will be at Triple Rock. And then we
And
all
do you need to have your own microscope set up in
Nope.
order to participate?
Nope.
No.
But we do ask that if you come and join in, that you support the establishment. We don't rent space, we don't reserve tables, and so come and be friendly. So there we are. There's just so much more to see. I'm not done. I mean, everybody is welcome to go, please. Thank you so much for coming. I'm just gonna
Keep it up.
Well, yes, we're just closing this meeting. Don't get us wrong. We're not done with you either, Janai, or with these microorganisms and plankton.
There's so much. It's so hard to do like 25 minutes on plankton or an hour on plankton. Oh, look at that. See, who are you? Driving the microscope is a lot like driving an Etch-a-Sketch. It's not got a motorized stage, so I have an X and a Y control, so I can turn it this way or that way. Oh, who are you? Hold on. There's some lovely dinoflagellates. A lot of times I find the longer I look, the more I find. So if you're doing research, you actually need to standardize the amount of time that you're observing a sample and the number of organisms like that you count and how you see them, how they settle. Interesting. Nice one.
Absolutely, dear. And if you're doing a Lakeside chat programming, you have to kind of standardize.
It's hard.
So I know. So sadly,
I'm saying good night. It was fantastic.
Thank you so
tonight.
much. Thank you, Janice, for all your great questions and support and everything. Thank you to everybody for joining us. This was cool.
such a wonderful talk tonight. Thank you, and thank you.
Yes. Thank you so much, Janai. Thank you to everybody.
There's just so much cool stuff.
Well, what time at Triple Rock? What did you say, 5 to 8?
Yeah, it's approximately 5 to 8. Yeah, because traffic and people getting off work. It's here. Let me.
And what was the other location? I missed it.
Oh, in San Francisco at the Valley Tavern. And if you join the San Francisco Microscopical Society, it's free for students. Then we post in our like member Slack, which is like it's an online forum for members who like microscopes and microscope stuff. And if we have to cancel or change it for any reason, that's where we post updates. So otherwise, it's a very informal, casual gathering, very relaxed. I found that we were doing a lot of events that were a lot of fun, but they tended to be really child focused. And a lot of our members enjoy that, but they also needed time to meet and talk with each other. And so this was kind of a preliminary attempt to provide that format for people. So I always bring 1 or 2 microscopes. And
Janai, we have a question about good microscopes for children. Do you have thoughts about that?
I do. It depends on the age of the child.
Of course,
who's asking? Sorry, I can probably actually look at chat now because I'm not driving a microscope.
Well, oh, go ahead.
No, go ahead.
Well, I wanted to say that your microscopical society sounds like bowling for nerds.
It's a very nerdy, cool thing. We've been around since the 1800s. So it depends on the age of the child, that you're and their level of interest. So many different types of microscopes. You can get things you can clip on your cell phone. You can get digital ones that are, handheld. If you're interested in seeing things larger, but not like going through the trouble of making slides and dealing with glass, like sometimes younger children, it's just not going to work. There's something called a stereoscope or a dissecting scope, and they are amazing. So you can put anything under it, and it makes it larger, and you can see more detail. You can resolve finer details. So you can put like your fingernails in your hands. You can look at fingerprints on your fingers. If you've got sweaty hands, you can actually like watch the little beads of sweat come up through your fingertips. Those are a lot of fun because you don't have to do anything, right? You can just stick whatever you want under it and make sure there's enough light and you can see it. If you have an older person who is, and by older, I mean middle school aged, who likes the idea of making slides and is interested in the process of how you go about seeing more detail and even closer, then a compound microscope, one that you would need a slide use like mine, but like a smaller non-toy version. And I bring one of each of those to hangouts usually, so people can play with them and see their actual microscopes. They're not toys, but they're less expensive. And then the society loans microscopes to members. So it's a perk if you're a member of the society, you can borrow one based on what's available, and you would need to be local enough to get it. We don't ship them. So yeah, it's a fun way to start figuring out like what kinds of things you want to look at.
So this is the San Francisco Microscopical Society?
Yeah, but I mean, we don't, you can live anywhere and go for our local events.
And the Triple Rock is a restaurant or a bar or something?
It's a bar, it's a brewery.
Oh, a brewery.
And they do serve food. So before a certain time in the evening, it's, I think, open to most people.
But you have your own space in there. It's not like there's a lot of noise.
Oh, there's a lot of noise. There's people. I take microscopes to bars. Yeah.
Okay. And what was the thing you just mentioned? I'm sorry, my name's Wendy, and I've really enjoyed this whole evening. And I've did you say a spectroscope? Is that what you said? A spectroscope?
No, sorry, I said a stereoscope.
Oh, sure, a stereoscope.
And it's a stereoscope because of a cool thing, right? So microscopes like the one that I use that magnify really with really high power magnification, higher power magnification, have a single objective.
And you have two then, stereoscope.
And a stereoscope has two. So you look with both eyes and you get like stereo vision. Like you can see things that are larger, and they were actually, my understanding is they're used a lot by people who need to do tiny manipulations of things. Because you've got that great vision, you can actually have a better
three-dimensional. Then it's more three-dimensional.
Yeah, and you don't need to really prepare anything. You just put it under it and focus. So it's a good place to start, but it's not going to be good if you want to look at slides of pollen or, diatoms. There's like knowing what you want to see is half the battle. Yeah.
So, but if you wanted to look at rocks, say,
stereoscope is a great place to start with rocks.
Where do you get a stereoscope? Probably lots of places, huh?
Yeah, they sell them on Amazon. I know Katie has one that she got recently that she likes. Right?
Stereoscope. Yeah. I actually, it was, I could find out about it. I didn't like it all that much. It was, I was looking for a cordless stereoscope or microscope actually, but it did have a low, a low magnification option. But it had very little space to actually put the specimen in.
Yeah.
You had to plug it in, and I didn't think it was really good for kids per se.
Yeah.
It was too hard to manage. But then, Janai, you lent me a larger one. Some of them are fairly reasonable. The stereoscopes are good. Yeah.
It's hit and miss. The society has a number of them that we loan out. So if it's something you want to try and you feel like joining, otherwise, the Amazon's a resource. Amscope is a generic kind of microscope manufacturer. They tend to not be very expensive, but the quality is like,
what you would expect.
Yeah, I mean, looking for a deal, right?
Right.
Be cautious, read the reviews, and
try it.
Find out what the return policy is.
A good idea. So I should sign off here, but I really have enjoyed it, and I will and Katie Noonan and David Woodward, thank you both for. I forget there's a third person, but she's not here, I think. But all of you make this happen, and it's really great. It's really great. Thank you.
Well, thank you,
Thank
Wendy.
you.
Thank you for all your great questions and for coming, and really appreciate our community that we have.
Thank you,
Oh
Wendy.
boy,
Really great. Yeah.
it is. It's
Thank
so.
you.
It's like what it's all about. I mean, it's just what it's all about. So thank you, Janai. And Southworth is your last. You're around here somewhere with that name.
Yeah.
Thank you all.
Thank you,
You're welcome.
everybody.
Good night.
So
Okay.
talking
Good
about
night.
Bluetooth microscopes for TVs. So I have one of my pet peeves is the software of the cameras that you use on microscopes that do that. I've never had a really great relationship with the software to be able to have it work smoothly. There's a lot of promises, like, oh, download this, and then you can, it'll be on its own Bluetooth thing and you can all see that. And I'm not impressed. I have not found one that has impressed me, but that doesn't mean they aren't out there. And if you find one, please share. But I use cameras. I like cameras that have an HDMI out with a clean HDMI feed if possible, and I can plug those into a projector or a computer or a monitor and access the images that way.
Thank you, everybody, so much. I think we should probably all say good night and start our weekend, and have a wonderful weekend, everyone, and hope to see you the next time. And continue. You can send me questions, and we can put together a nice post chat email to everybody.